So extraction was focused on the selective partitioning of a solute from a mixture or a complex matrix between two typically liquid phases or mobile phases in that case. And so the example that we used was a separatory funnel where you had an aqueous layer and some organic layer, toluene, and you mix those two things together and you shake it up really well so you have high surface area contact, and then you extract one solute into the other phase based on some specific chemistry. That could be acid-base character, that could be polarity, that could be complexation, etc. But that's how an extraction is designed. Chromatography is still separation, but instead of leveraging two liquid phases that are immiscible, now we're leveraging a stationary phase, a solid phase typically, or a gel phase that is then in contact with a liquid phase or a gas phase that's mobile. And so we have a mobile phase and a stationary phase here. I have a mixture of these green and blue compounds, and their selective interaction with whatever the chemistry is that's adhered to that stationary phase, they will either bind more tightly or more loosely, which then changes the rate at which they will pass through that stationary phase. And we call that elution, or the process by which a solute moves across or through a stationary phase is eluting, or the process of elution. And so that differential elution is simply based on whatever the differential equilibria are of binding to whatever the stationary phase is. And you can imagine this now, instead of designing liquid phases that are immiscible and can have some sort of selective affinity, now we design the chemistry into the stationary phase, where we design a phase that's maybe really polar, and that separates our red and blue based on polarity, or it could be based on size, it could be based on a number of things, as I'll mention here in a second. So if you've taken an organic chemistry class, probably the first two things that you'll experience as a budding chemistry major, or biochemistry major, or just scientist in general, are chromatography separations, either in a column or in a thin layer process. And so I have both of them sort of sketched up here, and I'll show you some animations that walk you through both. But let me just give you a quick idea. So in a column, which is this first picture, you can see what the column actually looks like over here. The column is just a glass tube with a stopcock, so it sort of looks like a burette, but it's really wide, and that accommodates our stationary phase. Normally the stationary phase is this stuff called silica gel, so it's SiO2, really small particles that have pretty hydrophilic capacity, so they're pretty polar. So you load that stuff up, which is the white stuff here, and you pack it in there nice and tight so there's no voids. And then you load your sample, which is a mixture here. This is the green little plug. And then you apply your mobile phase. In this case, the mobile phase is the thing that you are going to vary to sort of drag certain compounds through faster or elute them faster, and others stick. So that means typically you're changing the polarity of your mobile phase by just varying the solvent. It could be by adding pure solvents, or it could be adding mixtures of solvents in sort of a gradient mode. And so over the course of applying this mobile phase, it starts to preferentially pull through certain compounds that are less polar. The more polar ones are going to have stronger interactions, which is this orange band. Polar compounds are going to interact through intermolecular forces more strongly with that polar silica gel. Those that don't have polar moieties or functional groups are going to breeze through there pretty quickly, being pulled by the mobile phase, and that's how a column separation would occur. This is really common if you want to do workup of a synthetic reaction, and you want to do it on a preparative scale. And by preparative, I mean actually produce a reasonable quantity of a compound, especially if you're going to then subsequently use that in a latter reaction. If you're not concerned about preparative chemistry and actually working up your entire reaction mixture, then thin layer chromatography is really helpful. We call this TLC for short, and it works on the same exact principle, except now we have a really thin aluminum plate that looks like this. This is maybe 3 inches long by an inch wide. These vary in size, and then this thing resides in a beaker, and in the beaker, in the bottom of the beaker, we have a solvent which is going to act as our mobile phase. And then applied to the surface of this plate is the exact same silica gel, but it's been baked on so that it's sort of a thin cake instead of more of this like fluid-like structure that a column has. But everything else is the same. You, especially if you have a reaction that's running, and so you've got your round bottom and this thing's cooking, and you want to get a sense for how far the reaction's progressed. You'll often take little small droplets out at different times, maybe 10 minutes and 60 minutes and 4 hours, etc. And what you'll do is you'll just take a TLC plate and spot it, meaning you'll take the whole reaction mixture and put a tiny little drop right at the base of where the solvent will start to then act as a mobile phase through capillary action. So capillary action means the liquid's going to be drawn up the face of this plate in this direction. It'll be drawn up, and then it's the same but sort of inverse. In the column format, gravity did the work for us to draw the mobile phase down through the column. In this case, capillary forces are drawing or wicking the solvent up the plate. But the same thing occurs, so the more polar compounds are going to reside more strongly through a stronger interaction with the silica. The less polar compounds are going to proceed then up the plate. And that allows us a very, very quick and simple way to monitor the course of a reaction. Where are we in time? Do we have some of our starting reagent there? Do we have a mixture? Have we gotten rid of all the starting reagent? So it's often common then not to just spot your reaction mixture, but then to also spot, say, your starting materials. So that gives you a reference point for, how far the reference material moves up the plate. That'll tell you, okay, that spot is still my my starting material. Plates often look like this. There's ways to compute this. These RF values are just simply ratios of distance that a compound or a spot has moved relative to the solvent front, how far the solvent's moved. These are imaged at UV wavelengths, so this is 240 and this is 360. Often the reason for this is if you don't have colored compounds. So if you have colored compounds that are highly conjugated, then great, you can see them right on the plate. But often you don't, so you have these smaller, less conjugated compounds that maybe aren't aromatic, or maybe they are aromatic, but they don't absorb in the visible, and so you usually hit them with a little shortwave IR lamp that just lights them up, so you get to see some sort of absorbent. So this is one of my favorite cartoons of illustrating chromatography. It's actually, there's two of them I'm going to show you. So this would be column chromatography. So you'll get a sense for exactly how this works. So this is just a packed column. We'll talk about the differences between a packed and a capillary column in a little bit, but you've got a mixture of your products. Don't have a dump truck in real life, but those compounds, that mixture, or it could be one analyte in a mixture of a complex matrix. You dump those on, which really means you inject them onto the column somehow. That could be just using a pass-through pipette or pouring some on, or in certain cases it could be using a syringe. Now you administer your mobile phase, which then is going to preferentially, based on whatever those equilibria are between the stationary phase, draw those compounds through or elute them at different rates, and we can collect them off of the column. And so we call these fractions that we would collect off of the column. This is the basics of how a chromatography system will work. Okay, so and then a second video from the same maker. This is TLC. You'll see the resemblance in the video here. So here's our mixture of products, bunch of stuff. This is your reaction mixture, big brown mess that you just pulled out of your round bottom. There's your TLC plate. You can see a reference line drawn with pencil. That's a huge drop. Usually you don't want it quite that large. But that's okay. So you can start to see the solvent front moving via capillary action up. This is sealed just to prevent changes in volume of your solvent. So here goes the mobile phase. It's sometimes called the eluent, as you saw here. And then you can start to see the partitioning of all of the different colors. These are colored products over time, which gives you a sense for where you are in your reaction. So of course that column doesn't have to be made just of silica. Silica is really a general sort of stationary phase for a lot of organic separations, but when it comes to analytical separations and actually trying to differentiate certain compounds based on more complex chemical or physical traits, characteristics, we've got a whole range of different types of stationary phases, which generally categorize the different types of chromatography. So we could have interactions based on hydrophobicity. We could have things based on what we call this normal phase, which I'll talk about in a later video, as opposed to reverse phase. So we could have ion exchange type chromatography, which means that we're leveraging the charge and size of ions, meaning we have a stationary phase that's charged, either positively or negatively. We could do affinity type binding, which typically means you've got like antibodies or aptamers that have very specific type binding or recognition sites for whatever species you want to capture or elute, depending on how you design this. These are, of course, are much more expensive, but they're very, very specific. So they end up being used mostly in biological chemistry. And then we could have size exclusion, which is also a really common technique in biochemical methods when you're trying to separate out certain sizes of proteins or peptides. So you can construct your stationary phase to selectively adsorb molecules of a certain size or size range and partition out based on that. And so in like biochemical methods, you may use several of these techniques to extract out ultimately or purify a particular protein. It's sort of an art form because it really requires you to understand a lot about the species, your analyte that you're trying to extract, both terms in size, weight, morphology, you know, the tertiary and quaternary structures of these things, its charge, meaning where is it charged, at what pH is it charged, and when is it not charged, and also the polarity, so what do the residues of these things look like. So these are just some quick cartoons that I showed you, and then the ones over here show you the similar things. These are just pulled out of your textbook. There are more exotic stationary phases and types of chromatography that exist, but these are generally the main things that we exploit in terms of physical or chemical characteristics of molecules to separate. But really, in principle, if you can imagine something that differentiates a molecule from another, you could design a stationary phase that would selectively be useful for partitioning. It may not be cost effective. An example of that is there are columns that are utilized with enantiomeric purity, meaning if you want to extract only 1/2 of a racemate mixture that you produce in the synthetic reaction, you can design columns that are enantiomerically pure. Enormously expensive, but in certain cases when you're producing you know, it's especially in enantiomerically pure drug mixtures, it might be your best option. So really anything you can imagine to exploit a chemical or some chemistry or even physical properties, intermolecular properties of molecules, that could be designed into your stationary phase. So of course, the chromatography is simply just a separation. We still then have to be able to detect at least from an analytical perspective, all of the things that are eluting off of the end of a column. So if you've got a long column, you know, not necessarily preparative chemistry, but like a column and it's packed with some sort of stationary phase that's selectively partitioning out whatever these molecules are. An example would be an ion chromatography system, which we have at CC. These are fairly common. In that case, typically the stationary phase is made up of these beads, which are organic. Usually these are divinyl benzene, and there's fairly large microspheres, although they can be smaller. So you have this thing packed in here like this, and this is your column. And it's completely full. And then on the surface of all of these beads is an amine that's been linked, a sort of terminal amine, so that it comes out like this, and you end up having it can be an amine or an amide, but we usually call these quaternary ammoniums because they're protonated. So you have sort of a you know, an NR3 here plus at the surface of all of these beads. So in this case, the type of separation that you would be leveraging if you had a positively charged stationary phase is you would be separating probably negatively charged or anionic species in water. So this is a really common environmental application. So if you want to quantify things like sulfate, nitrate, nitrite, phosphate, etc., from aqueous solutions, which are, these are all present almost in all of these things, chloride, bromide, fluoride, etc. This is the type of column construction that you'd utilize. And so you'd pour your mixture on top, and then you would get differential partitioning based on that electrostatic interaction between your anions and the quaternary ammonium packing material. And so generally, this isn't absolute, but generally you would expect that the charge density on the ion would cause it to have a stronger electrostatic interaction. So that means the things that have higher charges on smaller ions are the things that are going to elute fastest. So the thing that you produce upon detection of your ion, so these things are going to elute off the column, right, we can't see these ions. So that means we have to design some sort of detector to actually measure what these things are, and ideally not just tell us when they come off, but also tell us how much. And so in this case, in almost all cases in an analytical separation, your separation front here, whatever this is, it's going to then be coupled to some detector scheme here. In the case of an ion chromatography system, what would our signal look like? Well, you've got a whole bunch of charged stuff that is coming off in bands, right? You've got, you know, a certain chunk of ions that are going to partition out there, and a certain chunk that's going to partition out there, and maybe, do I have another one here? And a big band that comes out here. So these things are coming off in waves, and what happens is you've got a bunch of ions that come off or elute in these highly concentrated bands. So what we do is design a detector that's simply just a conductivity meter. So you have two flat plates and some sort of voltage that's being applied to them. We measure the current, which allows us to compute the conductivity. Or, yeah, and typically that's the way it is, or we measure the resistance of the solution, and we expect that every time one of these bands comes off and comes through here, we get a spike in our conductivity. If you look at the curve here, this is a real ion chromatogram here. This is measured in microsiemens. A Siemen is the inverse of an ohm, which is a measure of conductivity. So what you're seeing are these compounds, and then on our x-axis we have time. So these things are eluting in this dimension. So we have things that come off the column first, which in this case would be like our yellow band. That would have been the fluoride in this case. And then we had chloride, nitrate, bromide, nitrate, phosphate, and sulfate. So all results of chromatography are always going to be called chromatograms, but the specific flavor of the chromatogram is going to be a function of the type of chromatography you did. So we could have a liquid chromatogram that is measuring maybe not the ions, because maybe we're separating a complex mixture of organics in your dirty reaction solution. In that case, our detector probably isn't going to be based on conductivity, but it might be based on absorbance. And so what's really common is to have just a lamp in here at the end, and we measure the absorbance at a particular wavelength or a couple different wavelengths, so that here, instead of microsiemens, we have absorbance. We could also have fluorescence here. We could have electrochemical current or charge, or virtually anything we've talked about thus far in this class as a signal. You could imagine that being coupled as a detection scheme on the chromatography setup. So the chromatography is simply the separation, but it's pretty much useless from an analytical standpoint unless we couple it to a detector. And so you can see here, you know, we've got fluoride coming off, which is, you know, this fairly small negatively charged ion, and then we've got chloride, and then nitrite, which would be, oops, not NO3, NO2 minus. You got bromide, nitrate, phosphate, and then sulfate coming off later. And, you know, it's not absolute, but there's generally some tracking here with the size and the charge. So the things that are larger and more highly charged tend to elute later, and things that are smaller and less charged tend to elute faster, simply based on the construction of our column in our stationary phase. Another really common type of chromatography, probably more common than ion chromatography, would be this thing called Hplc, which stands for, depends on who you ask, but either high pressure, which is technically the right name, or high performance liquid chromatography. We're going to talk a little bit in the next chapters about liquid versus gas type chromatography, but it really just describes the state that your sample is in when it's injected onto the column where the chromatography takes place. And so, as the name sort of implies, this is the separation of a liquid mixture, which makes it really perfect for things like water samples, but also things like reaction mixtures. So we have a couple HPLCs, maybe 3 of them on campus, one of which is the workhorse that gets used a lot. This is in our NMR room. And we actually have a UHPLC, which stands for ultra high pressure liquid chromatography, which we'll talk about in subsequent videos. But this is an example of some data that my students would have collected in one of our citizen science research projects, where we've been quantifying these things called PFAS, which stand for perfluoroalkyl substances. So they're perfluorinated alkyl chains, which are bad for your health, but they end up in all sorts of places, and then they bioaccumulate in living organisms. They're bad, and there's maybe a few thousand different isomers of these things. So they're difficult to separate because they may only vary by 1 branching carbon, etc. And so we use ultra high pressure liquid chromatography to separate these things out. And so what you're seeing here are a whole bunch of different PFAS molecules that have separated out. And this is a chromatogram, still with time on the x-axis. Here we're measuring intensity, and actually what we're measuring is the each one of these we're collecting a mass spectrum for, and we're actually summing all of the different ions that are being created in a mass spectrum for each of these. But that's just another detection scheme, which we'll talk about a little bit later.